Yep, that looks great.
Okay, fantastic. Well, it has been, it's amazing that it's been about 3 years since we met with this team and talked a little bit about Mouse Genome Informatics and the Alliance. And I think, as you'll see, the Alliance resource has really developed quite a bit since we last spoke to you. The purpose for today, what we're hoping to accomplish with this is to introduce or reintroduce to this team the Mouse Genome Informatics database and the Alliance of Genome Resources as a source of curated phenotype and disease associations for genes and alleles for using mouse and other model organisms. And we want to walk through a couple of examples of finding data in MGI and the Alliance that are relevant to the ClinGen evidence SOPs for gene to disease associations, and talk a little bit to you and get your feedback on ideas we have for building customized interfaces for ClinGen curators. One of the things that I think will be evident when Cindy walks you through a few examples is that we have a tremendous amount of data in MGI and the Alliance, but finding the relevant data to the needs of ClinGen curators specifically often takes multiple steps, multiple clicks through pages. And that may be time consuming and difficult then for you to directly find the information that is most relevant to what you're looking for. And so we've been thinking about ways to make that easier, and we would like to get your feedback on that. And then I wanted to end on some future directions as it relates to sequence variants. So, and the basic question here is, just like evidence from mouse models and other model organisms can be useful for evidence for gene to disease associations, is it possible that attributes of variants in model organisms could be relevant to the variant pathogenicity curation efforts of ClinGen? And we had some ideas on that, but we were hoping to get some feedback from this group on what directions might be the most beneficial. So for those of you who might not be as familiar with MGI, it's an expertly curated community knowledge base for the laboratory mouse. We are the authoritative source for mouse gene, allele, and strain nomenclature. So we work closely with the International Nomenclature Committee for the Mouse, and that international body develops the standards for nomenclature, and then MGI is the enforcer for those standards. We are the primary source of gene function annotation through the gene ontology, mouse phenotype annotation using the mammalian phenotype ontology. We're the authoritative source for mouse models of human disease, where the disease terms we use come largely from the disease ontology. And gene expression during mouse development is another one of the data types for which MGI is the authoritative source. So it's, if you think about, so MGI has been around, or components of MGI have been around for a long time. The database is actually updated daily, but those daily updates are packaged into weekly data releases, and the knowledge that's represented in MGI is associated to almost 300,000 references at this point. We have over 1,600 human diseases where there's at least one mouse model annotated to it, and over 70,000 mouse genotypes have been associated with phenotypes, observable phenotypes annotated through the mammalian phenotype ontology. I think one of the keys here is that we're very much a FAIR compliant knowledge base. So findability of information is really at the top of our priority. And the use of these ontologies as standard terminologies to help with data aggregation and data retrieval is one of the operating principles for MGI that I think make it particularly useful as a source of evidence for gene to disease information. So the other thing that's not often appreciated is that MGI is not a single database. MGI is actually a consortium of multiple mouse-related databases. So MGI is comprised of the Mouse Genome Database, the Gene Expression Database, mouse models of human cancer, several information resources and knowledge bases combine to form MGI. So we have quite an extensive range of data types that we integrate into this consortium. So the Alliance has been around for a couple of years now, but a lot of the time has been spent in sort of evaluating different annotations across model organisms and working on the harmonization of those data types. So it's a comparative genomics knowledge base. Right now, we include annotations from mouse, rat, zebrafish, Drosophila, worm, and yeast. And of course, comparisons always to human. And the data types that are the primary representation in the Alliance currently are alleles and variants, function, phenotype, human disease, gene expression, molecular interactions, and genetic interactions. The idea here is for the Alliance is to build a knowledge platform for model organisms, and we are extending the representation of different organisms in this. So Xenopus, for example, is one of the model organisms that we're working on integrating currently. So our goal is to be this one-stop shop for essential gene information across all the model organisms. And some of that is relatively straightforward. But as it turns out, there have been significant differences in how each model organism has solved a similar problem. So we spent quite a bit of time in going back and looking at how we've done annotation as individual model organism databases and harmonizing those processes and aligning the annotation so that they can be presented to the community in this comparative context. And Cindy will be showing you a couple of examples of the benefit of this harmonization process and now how easy it is to get phenotype and disease annotations for a gene across multiple model organisms without having to go to 6 or 7 different databases. So when we think about model organism evidence for disease to gene associations in the context of ClinGen especially, we think about it in terms of what our understanding is of your curation process and what kinds of information you are particularly interested in digging out for model organisms. And so, one of the things that we took from your curation SOP is, one line of evidence that you use is if there is a disrupted copy of a gene that shows a phenotype consistent with the human disease state. And so I think Cindy will show you a number of examples where the annotations that we provide and the representations can really help facilitate making this assessment very quickly with data integrated from many, many different publications instead of having to go to, say, PubMed and looking through one publication at a time and doing this assessment individually. The other type of information that we looked at when we were looking at your SOPs is this concept of rescue. And that is, if there's a knockout, can the phenotype be rescued by the exogenous wild type gene? We don't explicitly represent rescue data in the database. but we've been trying to work with some keyword searches of the abstracts that are associated with our genes that would enable someone to assess this information very quickly as well. And we're still working on that. But this is one of the areas that we don't necessarily support very well right now, but that we're trying to address. And so with that introduction, I'll stop and ask if there are any questions so far before I turn this over to Cindy, and she'll walk us through a couple of examples.
Does anyone have questions for Carol? Carol, I guess one quick question I have is just
Sure.
Regarding literature that is included in the database. How often do you update, and is it curated? Do you have folks like reading through those papers? For example, if you had multiple papers on the same model, how is that shown? And that's something that maybe Cindy will be showing us. I was just curious.
Yeah, I think when there's a publication about a new phenotype or something new to add to the annotations, that's certainly prioritized. And oftentimes, if there's a Me Too paper, a paper that has information, but we've already got that essential annotation in the database, on occasion those papers are indexed, but not, so they're associated with the gene, but they aren't curated in depth because they aren't adding anything new. And so I believe Cindy can comment on this, but our focus is always on adding the new information to the gene, and not necessarily indexing every single paper to a gene if it's duplicating annotations that we already had. Is that, Cindy, is that fair to say?
Yes, that's correct. I mean, we certainly prioritize anything that's a disease model that's a new, or something that's a mutation, a new mutation in a gene that we don't have any information for, as well as new phenotypes. But, if it's the 5th knockout allele and they're just studying something a little bit more in depth, we may not necessarily curate that paper, but we will associate it with the relevant genes and alleles so that if someone is interested in that further information, they'll be able to access it and find and be aware that it exists.
And we do, Jenny, so the curators are updating things every single day, but the releases are for MGI currently are on a weekly basis. So there are weekly data releases with the new information, new annotations.
That's great. Thank you.
Sure. So what Cindy's going to focus on and what she's going to show is a couple of different ways to get like the phenotype and disease information associated with the gene or a genotype. First from MGI, and there are really 2 main ways that one can access this information, and she'll show both of these. And then the Alliance, and as I said previously, the Alliance has come a long ways since the last time we spoke to this group, and there are some pretty nice comparative summary tools that the Alliance has implemented to enable you to look at annotations for a gene across model organisms. And Cindy will show you a couple examples of that. So Cindy, I'll turn it over to you and just tell me when you want me to advance the slide.
Will do. Okay, so when we were looking at how ClinGen was curating some of this data, we looked at a lot of your gene disease validity reports to understand what kind of evidence you were looking for, as Carol summarized. And so in the first case that I'm going to show you, you already have a definitive association between this gene ABCD1 and adrenoleukodystrophy, which is shown here on this page. Can I have the next slide, please? But you didn't have, at the time we checked, any information from the human, the model organisms. And so I'll show you how to get to that information in the next couple of slides. We'll start at MGI with the next slide, please. We use the Human Mouse Disease Connection portal to look for any associations to disease for the gene ABCD1. So when we search for that gene, the first view that you will see is a grid view for the human and mouse genes, and a summary of the high-level phenotype and disease associations. Now, what the squares mean is that if they're colored in, there's a positive association, there's an annotation to something in that particular category. If it's orange, there's a human annotation. If it's blue, there's a mouse annotation. And if there is a square with a diagonal with both blue and orange, it means there's both human and mouse data for that particular category. And the darker the color, the more annotations that we have for those particular phenotypes. Over on the left, after the gray demarcation, we have the disease associations. And in this case, we have both human and mouse disease associations to these particular categories. If you click on the cell itself and go to the next slide, please, you'll get a summary grid view showing the human annotation to the gene and several mouse models of the disease, adrenoleukodystrophy shown up on the top. So we do have mouse models of this particular gene that for adrenoleukodystrophy. If you go to the next slide. If you go back to the summary results for the HMDC and click on the next tab for genes, you can get more information about the human and the mouse genes, their genetic location, coordinates, the associated human diseases, as well as source information. And for the mouse gene, you can get the high-level phenotype categories that are reported. You can get references, including the disease-relevant references directly associated with the gene. And in the next slide, we also list the diseases associated with this gene, the IDs, alternate IDs, and the mouse models available that have been curated to this particular disease, as well as the references using these mouse models. Another way that you can get to some of this data is to go directly to the mouse gene page. If you go to our homepage and search for the ABCD1 gene, you'll get a link to this gene detail page. The relevant section for human diseases is a little bit down the page and includes all the associations to the human and mouse gene associations to human diseases, as well as a link to the mouse model table. Next slide, please. And if you click on that section, if you click on the link to the mouse models, you'll get a pop-up with the different genotypes that are annotated to this disease, the genetic background it was studied on, which is important because sometimes there's phenotypic variability depending on which genetic background in mouse that you use. Different inbred strains have different characteristics, and we've seen in many, many cases that disease phenotypes can come and go depending on which genetic backgrounds you're using in mouse. We have the reference association that supports that assertion to the mouse model, and the view button will take you to the genotype details for that particular mouse. Now, at the Alliance of Genome Resources, you can go directly to the homepage and search for the gene. We also have links from the mouse gene detail pages. If you click on the top link, the top right link, that'll take you directly to the Alliance homepage, where you can click on the Alliance gene page, which will take you directly to the mouse gene page for ABCD1 at the Alliance of Genome Resources.
And just to clarify, so at the Alliance, they're pulling all of the mouse information from MGI. So it's not that this is different information or coming from a different source. This is all still coming from MGI. It's just now being put in a context where you can easily compare the mouse to other model organisms.
Right. Yeah, so we're not, there's no information at least for mouse at the Alliance that we don't have at MGI already. So this is the summary section at the top of the gene detail page. And you can navigate to different sections of the page by scrolling down or using the left-hand menu to jump to different sections. It's a little bit similar to the MGI pages, except instead of section headers, you actually have a drop-down menu. If we go to the disease association section, which is the fourth one down, we can get to the ABC1 gene disease associations that are shown not only for mouse, but for all the other model organisms that are present in the Alliance. At the top, you see a grid ribbon view that provides a summary of the different model organisms that have been annotated, the gene orthologs. So at the top, we have the mouse because you're on the mouse page. The second is the human gene to disease associations. There's rat, Drosophila, C. elegans, and yeast in this particular view. Below the summary ribbon, we have a table of this information. The kind of association, in this case, we have is implicated in. We also have biomarker of, we have not implicated in. And you can see that the gene is implicated in this particular disease, adrenoleukodystrophy, the evidence, the source of that information. So for example, with a human is associated via both annotations at the rat genome database as well as OMIM provided by a rat genome database, zebrafish, as well as links back to the original data at the different model organism databases, and reference associations that support these assertions. So all of this information is summarized here. It's available for download if you click on the button on the lower left. And the different kinds of evidence are shown here as well, whether it's a traceable author statement or inferred from sequence similarity in the case of yeast, where they also did some cell work as well. So if we go to the next page, if you click on the link to the disease detail page at the Alliance of Genome Resources from the previous table, you'll get to a summary of all of the disease associations at the different model organisms databases shown if you scroll down this page, and we'll get to that in a moment. The top part is a summary, including the definition, any synonyms to that particular disease, references to other ontologies, as well as the sources of the annotations linked at the bottom to the individual model organism databases. Next slide, please. If we jump to the associated alleles section for adrenoleukodystrophy, you can see all of the different individual mutations that are associated with annotations to this particular disease and their associated references. And if you go to the next slide, these are the individual models, including the genotypes, including the genetic backgrounds that are associated with this particular disease. And importantly, we also do capture cases where a particular model does not recapitulate leukodystrophy. And the supporting references, so that if you're actually studying this other gene and you want to know what the differences are, you can go to that reference and to the annotations and see that this is not a model of this particular disease. Okay. So we showed you an example where there was definitive evidence. We're also going to show you an example from limited evidence. In this particular case, you had limited evidence to show that RAB28 cone rod dystrophy was associated with this particular disease. Sorry, I can't get it out today. Cone rod dystrophy 18. If we continue on, again, you have no model organism data associated with this. And if you go to MGI, to our human mouse disease connection, and search for RAB28, you can see that we do have an association, human association to this particular disease, as well as two different mouse models. If you continue on, the next slide, please, to our RAB28 gene detail page at MGI, and go to the disease summary section, we do have the link to 2 models associated with the disease, shown again here in the context of their genotypes, the supporting reference, and links to the phenotype view. And the next slide, please. In this case, I'm showing you at the Alliance, I didn't link to the mouse gene page, but I linked to the human RAB28 gene page. In this case, we have associations to the human gene from the original source from OMIM via the rat genome database, and the supporting information can be found at RGD. And the mouse gene association that we curated from the reference. So this is all available here at the Alliance in one shot. So the other thing I wanted to mention was that there are other cases, such as the RINT1 gene. ClinGen does not list a known relationship of this gene to familiar ovarian cancer, although you have an association to that in ClinGen. Mouse has mutations in this gene, but we don't list ovarian cancer as a disease, but we do have that as a phenotype. So there may be cases where you want to dig a little bit deeper as well and go to the actual phenotype to see if the mouse actually develops cases of ovarian cancer when we don't necessarily have an explicit disease annotation. So there's a lot more detail here that we could dig into, but that's not apparent in the disease associations. So I will stop here. Does anyone have any questions about MGI or the Alliance and the disease associations?
Have any of you looked at the Alliance to date as a source of information for gene to disease associations? There's a chat.
Yeah.
I will. So there's a message, Cindy, in the chat. We concern the experimental evidence for non-human models, but not the gene model association per se. So I guess.
Right. So yeah, so you're basically, you want to see the whole experimental evidence, and this is a way of getting at it, right?
Yeah, I think so. I don't know Xiaodong, if you want to elaborate on that at all, but I think what you pointed out, Cindy, about making sure that we're looking at phenotypes as well as just disease is really important. So that's a really good point. And I see that Christy said that she has used the Alliance. Christy, do you have any comments?
No comments. Yeah, I mean, we're really interested in if I'm glad to hear that some folks are accessing the Alliance, because I do think it can be helpful. And the real question is, does it make it easier for you to access the information that you're most interested in or most often access to do your particular curation work? And if it doesn't, we would certainly be interested in making modifications that would make it more useful to the ClinGen project. And so that actually leads me into one of the other things that we wanted to show today, or to talk about with you today, which is we've been working on trying out different ideas for customizing an interface to MGI that's specifically for ClinGen curators by streamlining access to the information on publications, the phenotypes, and the disease associations. And one of the things that would be useful for us at some point is to have you guys come and talk to our curators about what your curatorial process looks like. You've done a fantastic job of annotating that on your website, and you have all your documentation available. But that's really different than hearing it from an actual curator and what their day-to-day process looks like. And we would be really interested in that at some point down the road. But when we look at the SOPs and we think about, well, how would I try to find the information that's in this SOP using MGI? We have found that it does take quite a bit of navigation through the system to get to the information that is probably the most relevant. So if we go back to this example of ABCD1, if you go to the gene page in MGI for the mouse ortholog ABCD1, and you scroll to the bottom of that gene page, you'll see that we have 80 references associated with this gene. 6 of them are associated with descriptions of disease models. 36 of the publications are associated with mouse phenotypes. So if you have a mutation or generate an allele of this gene, and it's going to generate a phenotype, and there's 36 publications describing those. We have 25 publications describing the function of the gene and when and where it's expressed during development. So if you were to click on one of these links, you're going to be brought to a list of publications that looks something like this. It'll have the link to PubMed, the author, the title, the journal, the year, all the standard stuff, and then a toggle here where you could get a quick look at the abstract for this paper if it's available. And this is really helpful, I think. We also have all of the annotations that have been curated from this publication. So from this publication in MGI, there are 2 functional annotations. There's 1 genome feature, which would be the gene. There's a phenotypic allele. There's sequences, and there's a strain, a mouse strain described in this paper. And so if you looked at this reference and you wanted to get to this information in MGI, you can click, but you can't really see, you can't look at this representation of the reference and say, well, what are the phenotypes associated with the mouse described in this paper? What are the disease associations asserted by the authors, if there is one? You'd have to click through. So it's at least 2, if not more, additional navigation steps to really get to the information that we think is the most relevant to what your goals are for ClinGen. So we've been working on a modification of this that would allow you to quickly get to the publication and the abstract, but instead of having to do all of these click-throughs and navigate through different pages at MGI, instead we would provide right on this page a summary of the phenotypes and disease associations asserted in this publication, so that it was more condensed, less clicking around. You could enter a gene, get back the references with the annotation summaries right there in front of you. And then you could decide if you wanted to do more and maybe go and look at that paper for additional information. So one of our questions for you is, would a streamlined interface like this, where you could search by gene or by disease and get to the animal model information in such a way where you can see the reference and all the annotations, would that be helpful to your curation process? Would it be helpful to you? I think that's one of the questions we have. So I'll stop here and just ask for gut reactions and feedback. Is this missing the mark entirely, or would this really be of help?
So I have some thoughts, but I'm going to see if anyone else would like to has any ideas or thoughts on this, folks who do gene curation.
Hi, this is Lemuel. Thank you for all the information. Would it be helpful for putting in like the E15 or even a P0, the gestational ages as well?
You mean where at what point the phenotype was obtained?
Yeah, and all the other accompanying experimental evidence. I guess like if you're doing a whole in situ hybridization of some sort or protein extraction from what day, I guess could be helpful.
So I'm not sure we actually collect how much experimental methodology we pull from these papers and represent in MGI in a searchable format. If that is information, however, that is important to what ClinGen curators look at, that's important for us to know. So is that, you don't look just, oh, well, in mouse, this gene is associated with these phenotypes and somebody's asserted it's a disease model. You actually go in and look for specific things like how old the mouse was when the phenotype was observed and what methodologies were used to arrive at the phenotype annotation. Is that what I'm hearing?
Yeah, I know, like for me personally, sometimes I do a little more of a deep dive, and it's more, I guess, especially for genes affecting development is one where I could see that being used.
Yeah, I wonder if, it kind of depends on the disease area. So like Lemuel was saying, yeah, if it's a more of a developmental disorder versus like a metabolic condition or something, we may be sort of looking for different information. But I wonder, I think one thing I really like about this approach is that it kind of gives you a way to screen the papers, because especially for something like ABCD1, where there's, a ton of publications, it gives you a quick way to kind of look through and see which ones you might want to target, because as you've no doubt seen from our SOP, I mean, obviously we're not curating every single paper, but we're looking for the ones almost that give you the best bang for your buck kind of thing, which one's going to get the most points from, which are the best studies, which have really looked in enough depth to give as many phenotypes as possible that are related to this disease. So I really like that it gives you kind of a quick screen of the publications that are available rather than, just going to PubMed and kind of going through the list and, having to do all this work ourselves rather than, what you've come up with here. So I really like that. I personally feel like this would be really helpful, but again, you're taking Lemuel's comment about the ages at which phenotypes are seen. Yeah, I mean, I would say we take that into account just in terms of even thinking about severity of a disease as well. In humans, something may not appear until adulthood, but if it appears much earlier in a mouse, just like a day of age or something, that's something we would probably comment on. Do others have thoughts on?
Yeah, I mean, certainly we don't show that in this summary view, but when you get down to the genotype level where we have the annotations listed, we very frequently have the ages and the methodology listed in a free text note field. We just don't have it in a searchable format.
Right.
And we don't, we may have that information for the disease annotations, but we don't display age of onset at this time. So that's something that we can certainly think about for future improvements. So that's great to know.
Yeah.
That's information you want.
Perhaps even seeing the phenotype annotation here is, that's useful information. And then if this is a paper you're interested in, then kind of investigating that a little further. Be good. Great. Yes, so one thing I'm thinking about is just in terms of curators being aware of these resources. I mean, obviously that's one of the reasons that, we invited you to be here today, which is terrific. But one thing I'm thinking about as well, potentially, is including, maybe including links to these within our interfaces. And I think we have members of the Stanford team on the call, but we do this for some other resources. So for example, within our variant curation interface, we have links out to some of the splice site predictors, for example, but perhaps putting links to this resource from our animal models section. So I don't know what folks think. Anyone from the gene curation working group or Stanford team, thoughts on that? Well, it's certainly something that we can look into. And then also with reference to, trying this out and getting information back to you. One thing I'm thinking about is maybe just getting, any curators who are interested in maybe piloting this system, trying it out to give some feedback. And then we could come and talk with you all about that at some point. So yeah, that's an idea.
We would love that. I think, one thing we would really like to have like a reciprocal seminar. We'd love to hear, I think our curators would be really interested in hearing from you guys what your curation process is like. And I think that could spark some ideas and maybe some ways that we could better package the data to make it more useful for the type of work that you're doing. And then we would love to come back when we have this prototype thing worked up a little more robustly, to come back and show it and make sure that you guys know how to access it, and then just get feedback, just have people look at it and say, I would never use this, or yes, this is really helpful for me. So we would like to continue the discussion beyond today, obviously. And I guess, Jenny, we could do that through you, and you can connect us into the rest of the group.
Yeah, absolutely. I'd be happy to put out an invitation, and anyone who's on this call, if you're interested in participating in this effort, just let me know, and I can talk with our gene curation small working group as well. And we can just get a bunch of folks together, try it out, and then talk with you, because it just seems like such a fantastic resource, and I just want to make sure that folks are aware of this and the utility both for gene curation, and you'd also mentioned the variant interpretation as well, because I know there are some variant curation expert panels that are using mouse models. I believe at least cardiomyopathy. There may be others too that I'm just blanking on right now. But yeah, I mean, I think it's important to consider that as well. So
Great. Well, I'll talk about the variant stuff next. I'm glad to hear that. But there's a question in the chat. Can you please say to what ontology your phenotype terms are linked to? Are these free text terms? They are not free text terms. And Cindy, you want to talk about MP and maybe the connections between MP and HPO?
Sure. Yeah, so we use the mammalian phenotype ontology, which is what we had developed at MGI throughout for like the past 20 years. And it's also used by the rat genome database. We also have been working with the developers of the human phenotype ontology for quite some time to map our terms across these two ontologies. It's called the Unified Phenotype Effort, or UPheno. In addition to the mammalian phenotype ontology and the human phenotype ontology, other model organism ontologies are also trying to be incorporated and mapped across as part of some of the work we've been doing at the Alliance in conjunction with Monarch. Yes, it's an ontology. Yes, there's linkages to the human phenotype ontology, and in fact, in many of the cases, some of the terms are identical. But that work is continuing, and we'll keep you posted on the progress on that.
Great. So I wanted to sort of end by talking a little bit about the potential for using model organism variant data for variant pathogenicity curation. And so my reading of the ClinGen SOP 2.0 from January, I didn't see any real reference to the use of model organism variant data to help with this effort, and I think that's for good reason. I think it's actually probably a pretty challenging thing to do. But I think there might be cases where model variant information in model organisms could be useful to variant pathogenicity curation as well. For example, in mouse, for sure, there are variants discovered in human populations. Those variants, humanized alleles, are being created in mice to have a model to study that particular human variant. And the phenotypic and disease correlations when this humanization happens is often quite high. And so you might be, there probably are situations where the data that one can get from a humanized variant in a model organism could be relevant to the curation effort in ClinGen. And then we've also been exploring ideas for how to define variant equivalency across organisms. So, is it the same variant type? Does it have the same molecular consequence? What are there phenotypic consequences associated with it in model organisms? Does it occur in the same protein domain? It won't be at face value, there won't be a one-to-one correlation of coordinate spaces, but the consequence of the variant, the equivalency at that level of biology might be possible. So we've been thinking about this, but we don't really know if it's going to be relevant to ClinGen or not. But we have, at least in MGI, we have over 2,300 humanized sequence alleles. So alleles that have been created in mice based on a common disease variant in human or a presumed disease variant in human. So we have a lot of data related to this. And we've been working with our colleagues in the Alliance on translating a lot of these phenotypic alleles into sequence context using HVS notation, and then documenting the type and molecular consequence and so forth for these variants. So I think there's some potential here, again, to have model organism information assist in ClinGen curation efforts. But this is a very fuzzy area for me. And if there are ideas from this group on what we might be able to do together, or what we might be able to do to make the data more useful for you, we would be really interested in those ideas. So I just end, as we're getting close to the end here, I just want to thank at MGI, the leadership team and the executive team at the Alliance of Genome Resources. But none of what we're presenting of these amazing knowledge bases would be possible without the biocuration scientists and software developers who work on all of these model organisms, and the funding from our various NIH partners as well make it possible. So that's what we had to present about MGI and Alliance today. We are definitely open to any questions from you guys, but we're also interested not just in questions, but in feedback and your ideas. And we just put together a couple of questions here as well. So if you do use MGI or the Alliance, what's useful, what could be better? Does the comparative view of phenotypes and diseases in the Alliance actually help you, or is it not detailed enough? And one of the things we were interested in is for the genes that you guys are working on, whether there's disputed or insufficient evidence for gene to disease associations, how often do you revisit the model organisms for evidence to support or refute the associations that you're making? So thank you so much for the invitation, and we'll just open it up for discussion and questions.
Well, thank you both. This was just a terrific presentation, and just really exciting to see how much work has been done, and just such a fantastic resource. Just to answer your last question first, in terms of disputed or low evidence genes. So we do have a protocol within ClinGen where I believe we go back and look at those every couple of years to see if there is additional information that could change the classification of those gene disease clinical validity classifications. So yeah, so every couple of years we would be going back and looking at those. So that's now kind of standard practice. Yeah, but I mean, I'd be curious to hear from others on the call regarding, what you think would be helpful. And certainly, I think, the way I'm seeing it right now is just a start to, maybe having a more sort of focused project from ClinGen curators to really, try this out and give you more specific feedback and talk with your curators as well. I'd be very excited to do that. And in terms of, I think, the variant interpretation, personally, I mean, I only work on one variant curation expert panel, and we don't use mouse models. It's an enzyme deficiency. But I would be curious to talk with others who do use mouse models in their VSEPs, or maybe talking with the SVI as well regarding, which models are acceptable and how's the best, what's the best way to get that information. So anyone else have, feedback for Carol and Cindy right now, or as I said, also please do email me if you'd be interested in being part of an effort to, work on this and give them more feedback later.
There was one more question in the chat. Is search by variant using HGVS available in MGI?
Not yet. So the curation effort here. So it's a great question, and eventually there will be. Where we started with this is that, we have phenotypic allele representation in MGI going back, decades, way before there was even a mouse genome available. And so a lot of the allele information that had been in MGI was all curator notes about, the sequence context for a particular allele. And so it's just been in the past year or year and a half that we've started to go back and dig through that information and systematically curate it so that it has HGVS notation. We have, I forget, 60,000 or more phenotypic alleles that we have to work through. The team is doing a fantastic job, and we've developed some tools to assist in that curation, but the basic curation is still going on. So eventually you will be able to search for variants using HVS notation in MGI, or at least through the Alliance. So it's coming, and we'll keep this group posted as that functionality is released. And Laura, thank you. I'm glad that I hope that the Alliance is useful for you. I'm glad that somebody was able to learn something new about the resources today. That was our hope. And if you have any feedback at all, there's a number of venues for user support, both for MGI and the Alliance. So, did somebody have a hand raised?
That was an accident. I apologize.
That's okay.
Looking at the chat.
Okay, no worries. Yeah, I guess there was, oh, I had a question also for you guys. So for the revisiting model organism data for genes with low or disputed evidence. So we have ways to alert when there are changes, when there's new information available for a gene or a phenotype. So one of the things I was thinking about is basically taking all of your low confidence in disputed genes and putting them into this automated system where if there was a new phenotype or disease association in mouse for one of those genes, that we get an alert and then we could pass that on to you, or you could sign up to get those alerts. Is there a path for us to communicate easily with this team if we find information that we think would help with your curation activities or help update when there's new evidence available?
That's a fantastic question, and I think any alerts that we can get would be really super helpful. This is something that we could maybe discuss with our software prioritization group.
Great.
Yeah, so I can definitely bring that up there, because I know in the past we've talked about trying to get alerts, from other sources as well, like PubMed, for example, if there's a new paper. So yeah, I will bring that up with our software prioritization and see how that best be done, and unless anyone on the call has thoughts on that. All right, but or something like that could make sure that our curators are aware of this and how best to use the resource. So yeah, this has been really, really helpful. So thank you both so much, and we'll definitely be in touch about future work.
Great. We really appreciate the invite, and look forward to talking with you guys further.
Brilliant. Thanks so much. Have a great day.
Right. Bye.
Bye.